Hello, I'm David Pauza, Chief Science Officer of American Gene Technologies, a biotech company in Rockville, Maryland. Today I'd like to talk to you about our ImmunoTox program, which is an immuno-oncology effort focused on activating the power of gamma delta T cells to eradicate solid tumors. First, I'll give you a little introduction into the company background and just introduce you to our main areas of emphasis within AGT. So one of our areas is HIV disease. We've developed a cell and gene therapy for HIV disease. The IND was approved in August of this year, and we are moving into the clinic by the end of October. And we're very happy about this. This signals our initiation as a clinical stage biotechnology company, and we have tremendous hopes that this very innovative product will bring some relief to people suffering from HIV/AIDS. Phenylketonuria is a second area. This is an inherited disease that primarily is seen in young children. We screen all children born in the United States for the presence of phenylketonuria, and it occurs in about 1 in 12,000 live births in our country. It is an inherited inability to metabolize the amino acid phenylalanine, causing a toxicity that retards tissue growth and has a tremendously negative impact on development of the brain. AGT has developed a therapeutic vector for treating phenylketonuria. It's based on our lentivirus vector platform and hopes to achieve a one-and-done permanent cure that will eventually be available to young children to prevent them ever experiencing the symptoms of this disease. And finally, the gamma delta T cell program in immuno-oncology. This is, as I said, a lentivirus vector-based program to modify tumors and increase their susceptibility to gamma delta T cells. And today I'll give you some introduction to this program, some background to the concept, show you some data, and discuss the implications of this program as we're moving forward. So we term this ImmunoTox. And ImmunoTox describes a family of lentivirus vectors that we have developed based on our knowledge of how gamma delta T cells can be activated and directed to the purpose of killing solid tumors. So if you look on the slide here showing a model to demonstrate this concept, the model has green tumor cells at the upper left. These are unmodified, normally growing tumor cells. And we expose these tumor cells in situ, meaning in the tumor, to the ImmunoTox vector. That ImmunoTox vector converts a portion of the tumor cells that were green into the ones that now appear red in the figure. These are the ones now carrying a genetic modification that allows them to be potent for activation of gamma delta T cells. And we show that activation where a circulating gamma delta T cell, which is green, encounters that modified tumor cell and becomes an activated gamma delta T cell in brown. And that activated gamma delta cell can not only proliferate, but also have much increased ability for cytotoxic killing of tumor cells. Because those cells proliferate and can re-enter the bloodstream, they can spread to other sites within the body where they may recognize other tumors, even in the absence of genetic modification. This model also highlights our effort to overcome a principal barrier to the use of gene therapy for cancer. And that is, when you do a genetic modification of a tumor in a person, you only get a fraction of the tumor cells modified. And if your vector is intended simply to deliver a killing blow to the tumor, then you will only kill a fraction of the tumor and the rest will remain growing. In our case, we deliver a signal to the tumor that has a catalytic effect because it activates this potent immune response, driving the proliferation of gamma deltas and increasing their ability to kill all cells in the tumor, whether or not they had the prior genetic modification. So that's a quick summary of the ImmunoTox program. Now let's look at the details and the background to that program. What are gamma delta T cells? They're probably the least known of the peripheral blood T cells in human beings, compared to the more familiar CD4 or helper T cells and the CD8 or killer T cells. So gamma delta T cells are present in lower numbers, but as I'll show you in a minute, They have a very increased potency due to a special trick of how that population is created and maintained. The T cell receptor for gamma delta is, of course, the gamma delta T cell receptor. That's how these cell populations are named. And gamma delta T cells recognize what we call non-peptidic antigens. So they are not portions of proteins that are specific to some pathogen, like a bacterium or encoded by a virus. So CD4 and CD8 T cells recognize those peptides. Gamma delta T cells recognize something completely different, and their recognition is really based on understanding when normal cells have been stressed by the presence of a pathogen or by being malignantly transformed and becoming a cancer. Because those other cell types, 4s and 8s, recognize peptides, they need a presenting molecule to hold the peptide on the surface of cells while they look at it. In the case of CD4, it's MHC class 2 presenting molecule. In the case of CD8, it's the MHC class 1. In the case of gamma delta, it's a series of molecules called butyrophilins. In this case, a heterooligomeric complex between butyrophilin 3A1 and 2A1. Because of the nature of these presenting molecules and the very high complexity of the MHC class 1 and class 2 gene loci, CD4 and CD8 restriction, meaning how they recognize peptides, is polymorphic. And the meaning of polymorphic is that it's very different in each individual. So the CD4 and CD8 populations that arise in each individual are quite unique and never repeated among individuals. In contrast, butyrophilin 3A1 and butyrophilin 2A1 are simple, non-variable genetic loci, and presentation in this case is monomorphic. And the importance of monomorphic is that we all share gamma delta T cells that are very similar, if not identical in many cases, meaning when we find something that turns them on and directs them to cancer, we can use this repeatedly in a vast number of individuals. In fact, every healthy person in the population and most people with various kinds of diseases. So the T cell receptor repertoire, meaning what's the collection? In our terminology, the 4 and 8 are private repertoire, meaning they're not repeated in another individual. And as I said, the gamma delta T cell receptor repertoire is public, meaning you find it commonly, allowing us to use common stimuli for everyone. So how are these T cells made in the body? All T cells, including gamma delta T cells, start in the thymus. So they're derived from something we call double negative thymic precursor cells. And they don't have any indications of whether they're going to be 4, 8, or gamma delta at this point. The first thing those cells try to do is to make a productive rearrangement of the gamma chain of the T cell receptor. And rearrangement means these genes are quite complex and they have to come together in a very defined pattern, in a very defined sequence of events to put all the pieces next together to make a productive gamma chain. So productive means it's got all the components we expect to find in the final mature protein. If you make a productive gamma chain rearrangement, that cell can then go on to rearrange the second chain of the T cell receptor, which is delta, and produce a diverse population of gamma delta T cells, which are shown by the cluster of spheres there with many different colors. And the complexity is much higher than that, but we run out of colors when we're doing this kind of thing. That population then is quite distinct from the alpha beta T cell population, and getting to the alpha beta T cell population is in fact the default pathway if the gamma chain rearrangement fails. So in the absence of the gamma chain rearrangement, then you go on to make the alpha beta T cell. So once we have this complex population of gamma delta T cells, then we find that some of them, and I've marked those in red in the cluster of cells on the lower panel, some of them recognize a small molecule called a phosphoantigen, and we'll look at that more in a minute. Once they recognize phosphoantigens, those cells begin to be stimulated, to proliferate, and expand. And phosphoantigens are made in every cell in your body. Very similar phosphoantigens are made in bacteria. Production is elevated by many viruses, and they're also in other living pathogens like fungi or protozoans. So phosphoantigens are everywhere, and your body is constantly exposed to it. So over time, you begin to build up and preferentially select for those gamma delta T cells which recognize phosphoantigens. And that's really good for us, because those same T cells that recognize phosphoantigens are the ones we are targeting to turn into an anti-cancer response. So every person is already primed with cells that have the potential to attack their tumors. We just have to increase the ability to stimulate those cells at the right time and the right place to get them to destroy the tumor when we want it to occur. So those phosphoantigens, in this case, I'm showing you the human biosynthetic pathway that includes the phosphoantigen isopentenyl phosphate. And it's actually isopentenyl pyrophosphate is an alternate name, and that's why we label it IPP. Isopentenyl phosphate is in between something called mevalonate and the lower end of the pathway, which goes to isoprenoid synthesis on the right. And these are small molecules that modify proteins and cause them to stick onto cell membranes. And on the left, that same pathway can actually lead you eventually to things like cholesterol. So it's been known for a long time that inhibiting this pathway is therapeutic for certain diseases, including osteoporosis and related bone demineralization disorders. And that pathway can be inhibited with small molecule drugs called aminobisphosphonate, and these are quite commonly prescribed. Those molecules are competitive inhibitors of the enzyme farnesyl diphosphate synthase, FDPS. And when we block that enzyme, IPP has nowhere to go in the pathway, and it simply then accumulates. And the more it accumulates, the more likely it is to activate gamma delta T cells. So that I told you there was a metabolic sensor that gamma delta T cells use to recognize an aberrancy in the target cell, and this is way we can introduce it with chemicals. But of course, when you take aminobisphosphonate drugs, they're circulating throughout the entire body and having this effect everywhere, and they're not focused on cancer. So what's a different way to do it? If we make a viral vector, so we adopt the gene therapy approach to make a viral vector that delivers a nucleic acid inhibitor of FDPS, and this is what we call a microRNA, which attacks the messenger RNA for FDPS, reduces its level, and thereby reduces the level of the FDPS enzyme in the cell, we accomplish the same purpose. We reduce FDPS activity, we increase the accumulation of IPP, and we activate gamma delta T cells. But there's a very important difference. The drugs were systemic. A vector can give you local activation because we can put it only in the tumor or preferentially in the tumor. And so driving a local activation has tremendous advantages when you're trying to generate an immune response. To give you a simple and familiar example, when you go for your influenza vaccine, they give you a small injection in your muscle because you want the vaccine to stay basically in one place and generate a local immune response that later on will spread through your body. They don't inject that vaccine into your blood. If you inject it in the blood and it went everywhere, it would not be a vaccine. It would never trigger a productive immune response. So when you're trying to generate immunity, and that's your goal, local is always better. So here's how it looks sort of in lab-based data. And what these kind of experiments are is we use a tumor cell line called PC3, prostate cancer 3. That's a workhorse cell line in these kind of studies. And we can put the AGT401, which is one of the ImmunoTox vectors that downregulates FDPS and increases gamma delta T cell activation. So we put the 401 vector inside the PC3 cells by transduction. The PC3 cells are stably modified and stably suppressing FDPS production. Then we take those cells and we mix them in culture with normal human gamma delta T cells taken from peripheral blood. And we ask, does this modified tumor stimulate those gamma delta T cells And the way we know that they are being stimulated is that they produce a molecule called interferon gamma, and we can detect that. So on the right, you see that if you use a control lentivirus vector that has no anti-FDPS function, nothing happens. The lentivirus vector, that lentivirus vector plus ZA, which stands for zoledronic acid, and that's an aminobisphosphonate drug commonly prescribed under the trade name Zometa, that has some stimulatory activity on its own. The vector AGT401 has some stimulatory activity on its own, as you see in the day 9 purple triangles. But then you mix them together and the stimulation is really quite terrific when you combine them. It's far more than the sum of the two activations. It's a synergistic reaction. And you can see that again at day 19, where the zoledronic acid is still stimulating. The AGT401 stimulation has gone down a little bit by then, but when you mix them together, again, you get this very, very potent and unexpectedly potent reaction. So this tells us that the AGT401 vector is working exactly as we expected, and that it can be productively combined with something like zoledronic acid, where you have two ways to suppress FDPS, and sometimes that's better. And it's not always necessary, and we'll see examples later where it isn't necessary to use zoledronic acid, but in this case, for an example, it tells us that we're doing what we thought we were doing with the vector, and we're not doing anything else that is an off-target effect. So again, what are the advantages of using the vector over the systemic administration of aminobisphosphonate drugs if you want to activate gamma delta T cells to kill tumors? So the aminobisphosphonate are given by oral or intravenous delivery, and that's systemic distribution of the drug. The viral vector is given by intratumoral, it means direct injection into the tumor, or intravenous, because these vectors will find the tumors very efficiently if you inject them in the blood, if you inject them into the blood. And that gives you a local effect, where the modification is primarily in the tumor. The toxicity of aminobisphosphonate includes an inflammatory response and sometimes generates a very severe disease called osteonecrosis of the jaw. And the genetic modification is only going to be there as long as it takes for the gamma delta T cells to kill that tumor. Once the tumor is gone, the genetic modification is gone, the system goes back to normal. The potency of aminobisphosphonate for cancer therapy has been tested in a number of studies. And so you usually have to inject aminobisphosphonate, plus you have to inject a T cell growth cytokine at the same time. And what's been seen routinely is you get a ceiling of about 23% objective response across multiple clinical trials. And that's a good result, but not good enough. And we think by going to the local activation strategy for gamma delta T cells, we can increase the potency very dramatically. And this is the path on which AGT is on. So here's an example. The control animals who received the viral vector but never received gamma delta T cells are on the left. They were sacrificed very early because the tumors had grown to a size that requires sacrifice of the animals according to animal study protocols. On the right are the animals that were treated with both the vector and the gamma delta T cells. And you can see that most of them look fine, healthy, and lived quite a long time in the study, with only one that seemed to fail the therapy in this case. When we look at excising the tumor from animals, or looking in the place where we had put the tumor in the cured animals, we find the results that you see here. So the two left panels show you a tumor that had the viral vector in it from an animal that never received gamma delta T cells. And this is a normal, fast-growing, highly vicious tumor, as you saw on those previous slides. On the right two panels are the animals that received a genetically modified tumor plus gamma delta T cells, and you can see that all that's left is a fibrotic, scarred bed with really no evidence of tumor cells at all. So this was a very, very effective therapy. And to give you one quick note about how these experiments are done and why they're done in this way, it is because rodents do not make gamma delta T cells capable of responding to phosphoantigens. This is an immune response that's unique to primates and is actually best exemplified in human beings, according to my experience. So in the rodents, we have to use a human tumor because human gamma delta cells only recognize human tumors. So we use a human tumor implanted into those mice, and then we inject them with human gamma delta T cells. And you can see that this is an incredibly efficient way to get rid of a tumor in this model. And we are applying the gamma delta T cells when the tumor is already grown to a fairly sufficient size, making this a very stringent test of this kind of therapy. And you can see from the survival curves here that there's a small effect of the vector-only modification on increasing the survival of mice. But when you have the vector modification plus the addition of gamma delta cells, now you really have the home run outcome of most animals, and in fact, over 75% surviving with no evidence of residual tumor. So we've gone on from the vector 401 to start generating families of vectors that are more complex, address more steps in the process of gamma delta T cell activation and growth, and really try to ask how can we develop something that looks like it has the very highest potency with the fewest off-target effects that will be suitable for taking into the clinic as a therapy. Here's the vector 402, which downregulates the FDPS enzyme, as all vectors in this family do. But this one also produces a T cell growth factor, which is missing in mice because these mice have no immune system, and the mouse growth factors wouldn't work on the human cells anyway. So you can see here that on the right, we have 4 recovered animals that lived quite a while, and then one of the typical early sacrificed animals due to excessive tumor growth. Now, why do you only see 4 animals in this example? It's because when we made the first gamma delta T cell injection in this group, 4 of the animals were killed by that first injection because the gamma delta T cells attacked the tumor so vigorously that the mice died of something called tumor lysis syndrome. So that's a problem for an experiment like this, but it provides us a very important lead that we can manipulate the potency of this system by the addition of growth factors. We just have to be careful how we regulate them. And AGT is very far down the road of having the right controls over the amount of growth factor released to make sure that we keep the reaction local and we don't allow it to spread beyond that site, and we focus the immune response only on the tumor where we want it. Again, similar slide of the histology showing the abundant tumor growth in the absence of gamma deltas and the really massive wipeout of the tumor in the presence of gamma delta T cells. And these mice really, we can't find any evidence of residual tumor by microscopic observation. Another vector, 403, targeted a different part of the gamma delta T cell recognition system, here targeting the way gamma delta T cells recognize tumors that should be activating them. And so we made a 2-part vector that downregulates FDPS and carries this second factor. We put it into mice. Again, we had a really great survival outcome in those mice, showing that there's multiple pathways to the solution of how to make this vector more potent. And we have about 40 vectors in this family already, and we're moving forward to downscreen them and develop the small list of lead candidates. But repeatedly, we find very high potency, as again you see in a slide like this, very high potency, very efficient eradication of established tumors in these mice, and very few off-target effects because those animals that survived look just about as healthy as you can possibly look as a 100-day-old mouse. We can summarize those 3 experiments in these graphs. So the top panel shows you the decrease in tumor volumes once you start injecting the gamma delta T cells. The middle graph is the treated tumors are not plotted completely because the sample size went from 8 to 4, so we didn't plot that. But the bottom shows you the survival analysis where each animal is accounted, and you can see that there was a big drop in the survival of the blue curve animals for the 402 vector, but that was the tumor lysis syndrome. And then after that, all those the remaining animals all survived the duration of the experiment. So this is very encouraging. We have multiple ways to do this, and we're even building other specialized vectors that will bring more arms of the immune system to bear on the tumor to see how far we can push this technological development. Now, why are these things working so well? What's actually happening? Well, if you look at a negative control tumor, which is up on the left, and this is a tumor that was in an animal that was treated with the viral vector, 401, but did not receive gamma delta T cells. So it looks like the slides you've been seeing. And these sections were stained with an antibody to detect the presence of human T cells. So the upper right is a reference slide prepared from a natural human breast cancer, showing the stain working to detect human T cells infiltrating the breast cancer. Now, they did not make an effort in that particular experiment to classify these human T cells, but when we looked in our tumors, which is the lower right panel, then you see this abundance, massive abundance of infiltrating human T cells. The vast majority, greater than 95% of the T cells we injected into the mice were actually gamma delta cells. And we have done molecular tests on these tumors to show that to our level of measurement, 100% of the T cells you see here are gamma delta T cells. So you can quickly compare it to the natural cancer above and see it's highly infiltrated with T cells. And if you compared this slide to what's in the literature on gamma delta T cells, you'd see it's astonishing infiltration of these cells under the conditions that we've designed. So we feel very confident that this is a well-behaved reaction that we can move forward with, and we know a lot about how it's working and why it's working, including this rather phenomenal finding on the promotion of T cell infiltration into the tumor. Now we've moved on more recently to get away from simple model tumors like the prostate cancer 3 cell line to move into things more relevant to what we think is our primary target in the clinic, that being hepatocellular carcinoma. And there are lots of reasons behind this, and I won't go into those today, but we think there are very many advantages to the selection of HCC as one of our early targets to focus on with this ImmunoTox approach. So we began working with cell lines here, HUH7 and HEP3B, which are known cell lines representative of hepatocellular carcinoma. And we start by just characterizing them in the laboratory and show when we put the AGT401 vector in them, we get a dose response, reducing the amount of expression of RNA encoding the FDPS enzyme. So it's working as we expect. There's nothing really special about these tumors. HUH7 looks a little more susceptible. HEP3B looks a little more susceptible than HUH7, but we like the HUH7 because it grows more reasonably in mice and has a very predictable pattern of growth and expansion. So here we've done it now, and this is where you do a genetic modification of the HUH7 cells. You put them in a culture flask and you add human gamma delta T cells from peripheral blood. So you've mixed them together, and the HUH7 has been modified or not modified according to what you're trying to do in this experiment. And you ask, can you get that cell now to stimulate gamma delta T cell activity, again, by detecting the production of gamma interferon? And you can see on the top panel of 4 graphs that that is in the absence of zoledronic acid, the aminobisphosphonate. The bottom is in the presence of zoledronic acid. And the numbers in the upper right quadrant of each graph tells you how many cells, what percentage of the gamma delta T cells were stimulated, judged by the presence of gamma interferon. So just like before, there's a dose response effect of the vector alone. But here, even at a very low multiplicity of infection, which means a low input dose of the vector, you're already seeing very substantial activation of these cells. So unlike PC3, where the vector alone caused low activation, in these hepatocellular carcinoma cell lines, it looks like the vector alone is very potent. But it still responds to the addition of zoledronic acid, and we can still get these rather astonishing amounts of activated gamma delta T cells, reaching 70% of the cells in culture activated, as I've shown here, is really remarkable within this field of gamma delta T cell biology. We also know that we can deliver vectors efficiently to tumors through direct injection. So this is a viral vector that doesn't downregulate FDPS, but it makes a protein called luciferase. And if you feed it the right substrate, it will then light up and emit light, and you can detect it with a very sensitive camera. So you can see here that these tumors have been decorated, and all except for one mouse where it didn't take. We get light being emitted from the tumors, showing that we can get vector into the tumors. This also showed us we need to be careful about our technique and make sure we have really good delivery of the vector to the tumor so we reduce some of this variability. But we're moving along with all of these things to start really mimicking the clinical situation where we establish a hepatocellular carcinoma and we deliver the viral vector to that and follow with the pulse of gamma delta T cells. So here's a way to start looking at what might happen in tumors as we begin this mimicking of the clinical situation, right? So what's happening in these tumors is, now I'll show you this graph and explain it at the same time. So the blue curve is tumors that were modified with the viral vector and then put in and exposed to gamma delta T cells. I'm sorry, not modified with the viral vector and exposed to gamma delta T cells. So that's no genetic modification, gamma delta T cells. If I put the curve of the tumor growing in the absence of gamma deltas, it would be far steeper and to the left of that. Then the curves on the bottom right that are shown in red boxes and purple triangles are again putting in cells that are uniformly transduced with the vector, then exposed to gamma delta T cells in red, or exposed to gamma delta T cells plus zoledronic acid in purple triangle. But the cells in the middle, the brown and the green curve, show you something quite interesting. Here we took transduced cells, mean genetically modified, and we mixed them 1-to-1 with the unmodified tumor cells. And then we implanted them into the mouse. And we know that the modified cells grow much slower than the unmodified cells. So by the time we give the first gamma delta injection, that ratio has changed from 50-50 to about 20-80, with the lesser part being the genetically modified cells. So here's an example of what we think we could achieve routinely in a human tumor to get at least 20% genetically modified cells. And you can see that there's a very significant effect on tumor growth by the gamma delta T cells and a further enhancement of that by zoledronic acid, as we expect. So this is a beautiful demonstration of a dose response effect to the presence of the viral vector modified cells. It's showing you that you're killing tumor irrespective of whether it's modified or not. And it's getting us along the way to this first example of how we're going to start estimating dose, characterizing human tumors in the clinic, and really asking how this work is proceeding. And important to note that this is the vector that produces no T cell growth factor. If we then repeat this experiment, which is in progress now, to repeat this with a T cell growth factor expressing vector, we expect to see a tremendous push to the right of that 50% transduced group, meaning we've vastly increased the potency and overcome a barrier to tumor removal with these kind of doses of viral vector. And the survival curves bear this out. You have great survival of the two groups where you put in the 100% transduced cells plus gamma delta, and then you have a big shift to the right in terms of survival of the animals that had the 50% produced tumor. And remember, all of these animals are sacrificed usually according to a schedule or because their tumors get too big. And many of these animals that were treated in any way would have survived 50 days or longer, probably to around 100 days. So just to revisit the model one last time, we've shown you that we can produce vectors that are capable of making these green cells into red cells and making them able to stimulate gamma delta T cell function, that those stimulated gamma delta T cells do in fact come back and kill tumor cells, and they kill both genetically modified and unmodified cells, and in the body we would expect them to circulate, to find other tumors, including secondary sites and metastases that may have originated from the primary. So we think that this is a very important step forward in the development of our ImmunoTox program at American Gene Technologies, and we look forward to the next crucial steps leading up to our initial interaction with the FDA and finally getting this into the clinic. Thank you very much.